This lecture is part of a series of lectures for a course entitled The Physics of Diagnostic Radiology. The first lecture, MRI Systems and Hardware, is broken down into 4 parts. Lecture 1, Part 2, covers NMR and the bulk magnetization. At the end of this module, you should be able to describe tissues that do and do not produce an MRI signal, identify the 3 key properties of hydrogen that make it NMR active, write out the Larmor equation and define each term, recall where MRI is in the EM spectrum relative to other imaging modalities, and appreciate the role of B0 in generating so-called bulk magnetization. And finally, use your understanding of Zeeman splitting to explain why NMR has low sensitivity. So first we begin by discussing and describing nuclear magnetic resonance. It's important to recognize where we do not obtain an NMR signal from. In particular, the NMR signal or the MRI signal arises from both water and fat because water and fat contain an important NMR active nucleus, hydrogen. Hydrogen, of course, is found in lots of other chemical structures within the body, but the unique chemical environment of water and hydrogen in particular make them especially NMR visible. We find water and fat in lots of tissue parts of the body, including soft tissues like muscles, organs, and fats, etc., but also in fluids like cerebrospinal fluid, blood, and synovial fluid. It's important to recognize that we don't obtain MR signal from all parts of the body, and importantly, we don't typically see strong or bright NMR signals in hard tissues like cortical bone, ligaments and tendons, or the teeth. And we also don't obtain NMR signals from gases, such as the lung airspace, the sinuses, and the bowel. If you look at this image on the right-hand side, you can clearly see that the lungs appear quite dark, that subcutaneous fat and marrow inside long bones appears quite bright, and other soft tissues have varying degrees of signal intensity. The liver, for example, is distinct perhaps from the brain. And this becomes a strong attribute, as previously mentioned, of MR, that is, the ability to generate so-called soft tissue contrast. Remarkably, MRI can manipulate widely the underlying image contrast, and so in this particular image, you'll notice the blood within the heart is actually quite dark, and that's because of the specific pulse sequence or MRI acquisition that was used to acquire this particular image. So what exactly is an NMR active nucleus? Well, the NMR phenomena itself requires and relies on the fact that atomic nuclei can have spin, charge, and mass. And while charge and mass are generally more familiar to those that have taken a chemistry or physics course before, the idea of spin might be new. Spin is an intrinsic form of angular momentum. Nuclei have spin angular momentum under one of 2 conditions. They either have an odd atomic mass, that is the number of protons and neutrons, or they have an odd atomic number, that is the number of protons. Spin angular momentum leads to so-called precession, and we'll learn about this phenomena in the course of this lecture. But it's important to remember that spin and precession are not identically the same thing. The frequency of precession, what we call the Larmor frequency in MRI, depends on something called the gyromagnetic ratio. And this is a physical constant for different atomic nuclei, and it's unique for each of those NMR active nuclei. And so we'll find that hydrogen, while abundant in the human body and easily visible under MR, it's also possible to do imaging with other atomic nuclei, such as carbon-13, which also have spin, charge, and mass. So what is it that's so special about the hydrogen nucleus in particular? Well, obviously it's spin, charge, and mass, but it's other properties as well. In particular, it's high concentration in the human body and the frequency at which it precesses that makes it especially amenable to imaging. Here we see a large table of NMR active nuclei where the isotope H1 is shown on the top. It represents a spin-half system that's in high natural abundance. It dominates, it's the dominant form of all possible hydrogens. Importantly, the gyromagnetic ratio is 42.57 megahertz per Tesla. And this is a really important constant to recall if you're to become an MR scientist and to do well in understanding the following lectures. We define the hydrogen nucleus to have a relative sensitivity of 1, And given its natural abundance, we can calculate what we call an absolute sensitivity. In comparison, we can see that the H2, so-called heavy hydrogen, is a spin 1 nucleus. It has a very low natural abundance, a relatively low gyromagnetic ratio, and the consequence of having is that it has relatively low sensitivity or low absolute sensitivity. The relative sensitivity is at a constant magnetic field and an equal number of nuclei, whereas the absolute sensitivity is the relative sensitivity multiplied by the natural abundance. And so this is just the relative sensitivity here just notes or highlights for us that our ability to detect H2 with NMR is, a part per million relative to that of the hydrogen 1 nucleus. We can see that carbon-12, for example, has no spin and consequently is not NMR visible. But carbon-13, on the other hand, is a spin-half system that has and while it has a relatively low natural abundance, it is in fact NMR visible and can be used in a variety of different experimental conditions where carbon molecules are made with carbon-13 and become NMR visible. Now, without going through the rest of the table, I'll leave that to you to do under review on your own time. You'll notice that there are other NMR active nuclei, each of which has unique attributes and are pursued, at least in the research context of MR imaging. So here we begin a better description of the underlying NMR phenomena, and we begin by looking at a bunch of water molecules. And a water molecule, of course, is comprised of 2 hydrogen nuclei and an oxygen. And we can zoom into the hydrogen nucleus itself, which we know is a proton, or H1. That proton has a positive charge, and as we mentioned before, in addition to having charge, it has so-called spin. Charge and spin combine to produce what we call a magnetic moment. That is, the hydrogen nucleus actually behaves like a small bar magnet, and when placed in a large external B0 field, will align with that external field. In addition to charge and spin, we have spin and mass. And spin and mass, just as in a spinning top, produce so-called angular momentum. Now, a top will both spin and precess in the presence of a gravitational field. The hydrogen nucleus will spin and precess, spin natively, and it will precess in the presence of a B0 field. So the fundamental consequence of having charge, spin, and mass is that the hydrogen nucleus precesses at a particular frequency that we call the Larmor frequency. And the Larmor frequency itself is governed by the gyromagnetic ratio that we saw in the previous chart and the externally applied magnetic field, in this case designated as B0. Now we'll learn in MR how important the Larmor frequency expression is because it dictates or tells us rather that the precessional frequency of the hydrogen nucleus depends on gamma, which is a constant, and not strictly B0, but any and all applied magnetic fields. And MR relies on using the superposition of multiple different magnetic fields to do things like spatial encoding and consequently form images. It's instructive to recognize where MRI lies in the electromagnetic spectrum. If we think of other imaging modalities such as X-ray and computed tomography, mammography, and even PET, these are typically, or not typically, these are in fact very high frequency imaging systems and consequently very short wavelength imaging systems as well. So the wavelengths are on the order of 10 to the minus 10th meters, very far on this left-hand side, if you will. We can move through other imaging modalities that might include ultraviolet or visible light systems, finally infrared systems, and even millimeter wave scanners that are used now in airports and so forth. Until finally on the right-hand side, we see magnetic resonance imaging. Magnetic resonance imaging actually does imaging in a relatively low frequency domain, the megahertz or tens of megahertz regime, and also has relatively long wavelengths on the order of centimeters, tens of centimeters, something like this. And so we recognize MR as being on the far right-hand side of this electromagnetic spectrum and quite distinct from most other imaging modalities. We'll now turn to describing the so-called bulk magnetization. The bulk magnetization just refers to the idea that any imaging voxel or small volume, if you will, contains billions and billions of water molecules and hydrogen nuclei. And while we refer to each of the individual hydrogen nuclei as a so-called magnetic dipole, the ensemble behavior of billions and billions of these magnetic dipoles is referred to as the so-called bulk magnetization. Now, the individual magnetic dipoles are randomly oriented under equilibrium conditions where there's no applied external magnetic field. But as we'll learn, there can be an organization imposed of these magnetic dipoles such that a coherent bulk magnetization forms. The bulk magnetization itself is a vector field quantity. It has a magnitude and it has a direction, and the direction that the bulk magnetic field or the bulk magnetization points along depends on the applied external magnetic fields. So really, we just consider that the bulk magnetization itself is the ensemble or sum behavior of perhaps billions and billions of individual magnetic dipoles. If we think about a typical imaging voxel in MR, MR has modest spatial resolution. So in a pixel that has dimensions of 2 millimeters by 2 millimeters by 10 millimeters, then we see that this is associated with 10 to, approximately 10 to the 23rd magnetic dipole, so billions and billions of spins. Now, MR isn't sensitive to getting imaging information from each of those individual dipoles, and we'll see later why this is the case. So in the presence of no externally applied field, that is, the B0 field is off, the individual magnetic dipoles, here represented as bar magnets, are randomly oriented in every possible direction. So the spins point in all directions. And this diagram here is just meant to represent the scanner bore that a patient may lie in. And we typically associate the long axis of the scanner with the Z axis and the transverse directions with both X and Y. So with no B0 field applied, the spins are going to point in all directions. We can turn the B0 field on, and the B0 will polarize the spins and generate so-called bulk magnetization, such that the magnetic dipoles align with the magnetic field. Now, in point of fact, the magnetic dipoles do not uniformly align along a single direction, that is north pointing up and south pointing down. In fact, we end up with two possible configurations, the so-called spin-up state and the spin-down state. This is a quantum magnetic consequence, and energetically, either of these states is possible. Now, there's this phenomena called Zeeman splitting, and Zeeman splitting just describes the distribution of spins in the spin-up state, the number of spins in the spin-up state, minus the number of spins in the spin-down state, divided by the total number of spins. So this is something like the percentage of spins in the spin-up state, but we'll see we're far from percentages, but rather in the parts per million range. And the distribution of spins between the spin-up and spin-down state is equal to this constant on the right-hand side that depends on the gyromagnetic ratio, Planck's constant, the externally applied B0 field, all divided by Boltzmann's constant and the external or sample temperature. So if we look at a simple example of what the Zeeman splitting proportion will be, we can use the according constants. We could choose a temperature, say room temperature. You might consider body temperature as well, depending on if you're imaging a chemical sample or a human subject or an animal subject. And then we can choose an externally applied field. So in this case, we choose a 1.5 T field. And what we find after solving this equation is that we have about 4.5 parts per million difference between the spin up and the spin down state. And that is to say that there's, given that we have a very, very, very large number of spins, it's almost 50-50, right? It's almost half the spins pointing up and half the spins pointing down. And the difference in population between the spin up and spin down state is only about 4.5 parts per million. So there's really only a few part per million bias, and consequently, MRI has relatively low sensitivity. We're not detecting signal from all possible hydrogen nuclei. We're detecting signal from the difference in the population between the spin up and the spin down state. Now, if you look at this expression, there's really only a couple things that can change because most things are constant. So we can change the external temperature and we can change B0. Now, changing the temperature may or may not be practical. We could go to lower and lower temperatures and image chemical samples, for example, at lower temperatures, and that could improve the overall sensitivity of the experiment. Or alternately, we could change and increase, for example, the B0 field strength. And this expression, the Zeeman splitting expression, is what primarily drives, or at least in principle drives, an interest in higher and higher magnetic fields for MR imaging. And so while 1.5T was the earliest clinical standard, and that moved to 3T for lots of applications around 2000 or slightly before, now people are increasingly investigating 7T systems, 9.4T systems, and even higher whole body systems for human subjects imaging. We'll talk about the complexities associated with very high field imaging in later lectures, but this is one motivation for considering higher field strengths. Thank you. That concludes this lecture, and we'll return for the next part in a later lecture.